Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm very excited to be here today with Frank Gentile, who's the Chief Executive Officer of Casma Therapeutics, and he's leading their efforts to develop cutting-edge therapies that are focused on autophagy and tackling inflammatory, neurodegenerative, and lysosomal diseases. So we're going to get into all those areas today, and also where they overlap in some diseases that I know everybody will have heard of, like Parkinson's disease and Gaucher. Before joining Casma, Frank was a venture partner at Third Rock Ventures, and I think you'll be able to tell the story when we start, but you all incubated and really started this company from day one, and you've been going at it for a number of years and have some really exciting data and I think vision and mission for the future to share today. So Frank, thank you for joining us and really looking forward to it.
Thank you, Patrick. I'm really excited to be here. We're very excited about our TRPM1 program, which we sort of announced to the world last week and presented the data at the GBA1 conference in Montreal.
As I mentioned to you before we were recording, I had some teammates that were in the audience for that and saw the talk. So again, congratulations to you and the team. I'd love to go back to the origin of this, actually. So maybe you can talk a little bit about how you and your colleagues at Third Rock came into the idea in the space and tell people a little about this pathway and what the insight was that led you to start Casma.
Sure. Let me start by saying what the pathway is and what it's not. And so around the time that we started Casma, one of the things that people were pretty excited about was the entire target degradation field. So instead of inhibiting a target to downregulate its activity, could you just remove the target from the mix? And that's where companies, focused on PROTAC or proteasomal-based degradation sort of came into being. And that became a pretty exciting area. Now, the proteasome is one of two targeted degradation processes in the cell. It's specific to things like peptides or proteins, not particularly large targets. And so, for example, if you have a mutated KRAS, the proteasome might be perfect to remove that mutated KRAS. Okay, the autophagy system is the other housekeeping process of the cell, but it's specific to much larger cargoes. So by larger cargoes, what do I mean? Dysfunctional mitochondria, large protein aggregates like alpha-synuclein or tau, oncogenic or inflammatory signaling complexes like the mitosome, and things like lipid droplets, which could be important in obesity. The autophagy system is perfect for that. And so back when we were incubating the idea of a degradation company inside of Third Rock, the autophagy system came up as the most versatile of these two degradation systems because of the number of targets that you could degrade. And at that time, there was emerging science about more and more about the importance of autophagy. And while we were incubating Casma, in fact, and we brought in probably the two best autophagy scientists in the United States, Beth Levine, who discovered the first autophagy mammalian gene, Becklin-1, and also Skip Virgin from Washington University in St. Louis, who was a large researcher in the autophagy and immunology area. At that same time, we were beginning to incubate Casma. I think the world woke up to sort of autophagy because the Nobel Prize in 2016 was awarded to the man who discovered the first autophagy genes in yeast. This is Yoshinori Ozima in Japan. And so that became a place where companies got very, very interested in this pathway. And so it actually helped us in our incubation period, when we and to the launch of Casma in 2018.
That's a super helpful breakdown, and I think it helps me at least position where PROTACs and proteases play and versus the autophagy side. So Maybe you could talk us through some of the diseases that people may have heard of that this system is involved in, but also some of the rarer one. I mean, there's a very long tail lysosomal storage diseases that I think you and your colleagues are also thinking about. Maybe you could you could help expose that a little bit.
So obviously lysosomal storage diseases are extremely important in the autophagy system because you recall that the way the autophagy system works is that a cargo is identified. Okay, that cargo could be an aggregated protein, an organelle, a lipid droplet, etc. A cargo is identified. The autophagy initiation genes, proteins, go encircle that target and create a double membrane. That's called an autophagosome. So that's basically the equivalent of the proteasome. It's a way of sequestering the target. The autophagosome then migrates to the lysosome, and merges with the lysosome. The lysosome contains all the important digestive enzymes to either degrade the target, and recycle those components into the rest of the cell for use, or it takes the target and brings it to the cell membrane and deposits the target outside the cell in a process called exocytosis. So that's overall how the autophagy system works. So there's many diseases where the autophagy system has been implicated. And for example, any disease where you're clearing a large target involves the autophagy system. So almost all neurodegenerative disorders, Huntington's, Parkinson's, Alzheimer's disease, are all diseases where the autophagy system is critically important. But it's not just there. NASH, as an example, is important in the autophagy system with the ability to clear lipid droplets. Things like specific B-cell lymphomas that have mutated MIghty88. What mutated MIghty88 does is it causes the permanent aggregation of a complex called a mitosome that is involved in oncogenic signaling in B-cell lymphoma. And in fact, there's a subset of B-cell lymphoma patients called Waldenstrom's patients that have this mutated MIghty88. And that is a large oncogenic signaling complex that can't be degraded by degrading a single protein the way the proteasome does. So it's involved in metabolic disorders, neurodegeneration, renal disorders, liver disease, obesity. There's almost no part of the body, and obviously lysosomal storage disorders, there's almost no part of the therapeutic landscape that autophagy does not play some kind of role.
When people think about Parkinson's and think about the lysosomal storage disorders, it's often about GBA and GBA1. It's about the GBA1 pathway because the biology is really well understood there as to be aligned with what you're just talking through. But is it actually an issue in, you mentioned Parkinson's, Alzheimer's, Huntington's. It strikes me actually that so many of the brain disorders are involved in this in particular. And I'm curious about whether that's coincidence or why so many of these brain disorders do involve these really large proteins. But also, what's the story with Parkinson's? Is it everyone, or is it just particular genetic subtypes that are in this category?
Patrick, that's a great question. And let me go back to the lysosome for a second. Why are people focused now on the lysosome for diseases like Parkinson's or Alzheimer's, and not just sporadic Parkinson's, but as you said, GBA Parkinson's, Parkinson's associated with LRRK2 or TMEM175 or ATP13A2? Why are people starting to focus on the lysosome? It's become very clear right now that the lysosome plays an incredibly important function in these diseases. I'll just mention one right now, but there are others. One is that in these diseases, there is dysfunction of lysosomal acidification. This is critically important in degrading the cargo, the substrates. And so one of the things that people have been looking at is how do we restore the function of the lysosome in these diseases? One way to do that is our program, our TRPM1 program. One of its key things it does is acidify the lysosome. And so regardless of whether your mutation is GBA, LRRK2, TMEM175, et cetera, what's critical is that the lysosome needs to be acidified, and TRPM1 does that. Another thing that's really important, and this is specific to GBA, what's clear is that if you're a patient and if you're a person and you're heterozygous for GBA1, you may not be symptomatic. You may never get Parkinson's symptoms. Okay. And there's emerging data that there are a number of genes that act as genetic modifiers for people who are heterozygous for GBA1. Okay. Many of those genes are genes that are lysosomal genes.
Right. That makes
And so,
sense. Yeah.
so what's emerging right now is that GBA1 patients are symptomatic often when they have not just a GBA1 mutation, but they also have this second hit in a lysosomal gene. And in fact, our TRPM01 program has shown that if you have a model like this, and the specific model are animals that have a GBA1 mutation, and they also have a mutation in ATP13A2, which is a common lysosomal gene. Those animals develop Parkinsonian symptoms. And because TRPM01 acts at the level of the lysosome to reacidify the lysosome, and it does other things. It promotes exocytosis. It helps with the assembly of the VATPases, and these are critical for cellular metabolism. And it also increases lysosomal biogenesis, so makes more lysosomes. And finally, it increases autophagic flux. Because TRPM01 does all these things, regardless of whether your second hit in GBA is ATP13A2 or other second hits in a modifier gene, we think TRPM01 could be an extremely valuable program for these patients.
That's fascinating.
Which is also true in sporadic Parkinson's, by the way, this lysosomal acidification issue.
Where my genetics brain is going to that is thinking about how you can use either, a handful of genes like the known modifiers, but also polygenic scores to collapse onto these pathways and say how many hits or what kind of burden does this patient have on the path. And I suspect that you may find something really interesting in sporadic patients as well, right? That it does point at that biology.
And Patrick, later we can talk about a little bit about our, the efficacy studies that we have in preclinical models. But we have efficacy studies in Gaucher's models, GBA1 models, but we also are able to clear aggregated alpha-synuclein in an animal model, and that's more of a idiopathic or sporadic Parkinson's model. So there is something still about that lysosomal target that's important in sporadic PD. And we have data to support that.
That's interesting. Well, let's talk about that now because I'm curious of what you saw in the preclinical models and how you thought about where to go with this first, because clearly you had a lot of options in the direction to take it.
Okay, well, let's start with sort of the most robust of the most difficult and maybe robust of these models, and that's the CBE model. So, and we talked about that at the conference last week. So CBE is a small molecule. It's an irreversible inhibitor of GCase, right? And so GCase obviously is implicated in Gaucher's and Parkinson's, and there are a number of companies out there that are trying to activate GCase. And if you treat animals with CBE, it's a really rigorous and difficult model because over a short period of time, animals treated with CBE die of CNS pathology. So what have we done? We treated animals with CBE and we treated them with our TRPM1 agonist. And those animals, first of all, don't develop Parkinsonian symptoms and have significantly extended lifespan. Twice the lifespan of the CBA animals alone. Wow. And so this is a really robust and very, very difficult model. One of the things we also did is, and this was also shown at the conference last week, is we treated these animals with our development candidate, CSM101, and we also treated it with a different molecule that is very structurally similar to CSM101, but 100-fold less potent on TRPM01 on the target. And by the way, TRPM1, as I should be clear, is a target that sits on the lysosomal membrane. Yep. When you agonize TRPM1, you increase calcium flux across the membrane. And so there we saw that the less potent TRPM1 agonist didn't really ameliorate the symptoms or increase the lifespan, whereas our TRPM1 agonist did that. We did a similar study in a GBA1 knock-in model, where we saw significant reductions in inflammatory markers in microglia and other inflammatory cells in those same animals. We did a GBA plus ATP13A2 double hit model, which is a really strong model for GBA Parkinson's because of what we just talked about, having that second hit in a potential modifier gene. And then finally, we did a model of alpha-synuclein clearance. And here you take human alpha-synuclein in an AAV vector, you put it into the brain of a rat, it develops alpha-synuclein aggregates, and then we were able to clear those aggregates with our TRPM1 molecule. This is considered a reasonable model for sporadic or idiopathic Parkinson's disease. And so one of the things that you might be asking is, why all these models, and there are no great models for these disorders, Patrick. Yeah. There are, and so to be rigorous, you have to actually try a number of these models and sort of develop this sort of cohesive picture of what is happening. And so now we think we know what is happening. TRPM1 acidifies the lysosome. It promotes exocytosis. It promotes the making of more lysosomes. And finally, it helps aggregate the VATPAs, which is important in cellular metabolism. And so we think, by the way, we've published on this also in Science Advances in 2021. So we think, we've put the time in now to really understand how this target really operates.
What is the landscape of non-animal models in preclinical? Are there good organoids or anything like that? Or is that coming in the future? Or is this just really hard to model in a kind of cell in a dish sort of paradigm?
There are just no good organoids for these things. The only other models that are out there are sort of iPSC models, which are notoriously difficult to deal with. But there are no organoid models here.
I mean, it seems like you put together a good enough data package to start to move into humans. So I'd love to hear about what you're hoping to learn there from some of the early studies, and if you're able to share anything about what the high-level plan. Anyone who's done these sorts of programs know that as soon as you come up with a plan, it changes. But your best guess at what it looks like for the next year or two would be really interesting.
Sure. We actually think we have a super exciting, clinical plan here. But and I'll start actually with the clinical plan and then tell you after what we expect to get out of that clinical plan. Right. And so, after IND filing and approval, the plan would be to do a single ascending dose trial in healthy volunteers. We're planning approximately 5 different dose cohorts. We're not sure we're going to need all 5, but that's the plan for the single ascending dose. We're going to switch from there to, of course, the multiple ascending dose. The first cohort only for the MAD portion will be in healthy volunteers. Then we'll be switching to Gaucher's patients who have Parkinson's disease.
Wow.
In the MAD component there. And that will be a placebo-controlled MAD component. There'll be at least 2 doses, approximately 30 patients per dose, 20 on drug, 10 placebo. And then we'll offer the placebo patients drug in an extension part of the MAD trial. So it's sort of a phase 1B, 2A design. Yeah. And people who are working in this field, are more and more using this kind of design. Why GDPD patients? Well, there are a number of reasons. First of all, it is a massive unmet medical need. These patients have Gaucher's disease. They often develop Parkinson's disease. They are excluded from nearly every other Parkinson's clinical trial because they have Gaucher's disease.
Right.
It's a rare disease. It's an unmet medical need, but it's also a well-defined patient population. Why is this important? Because these patients, they're Gaucher's 1 patients typically. They're also taking Cerezyme, which is the ERT for Gaucher's patients. Cerezyme doesn't get into the CNS. So a drug that gets into the CNS can help them with their Parkinson's symptoms. But the other thing here is that these patients often have residual disease in the periphery, even though they're taking Cerezyme, because Cerezyme, for example, doesn't really help their bone pain. Yeah. So we think after 3 months on drug, you could actually get a signal in these peripheral markers, especially bone markers. We also think that in the lipids, you can get a signal in the CNS, by looking at these lipids, which are the toxic components for these patients. And so we're pretty excited to be able to get a clinical signal in about 3 months' time versus 6 months or a year in these patients. Yeah. So that's the plan going forward. So what do we expect to see? Obviously, safety. Obviously, we want to pick the 2 clinical doses. for the MAD component, but we're going to be looking at these peripheral markers, specifically bone, and we're going to be looking at CNS markers, specifically lipid in those patients. And as a biomarker, we believe that not only this will be important then for moving into a trial for the GDPD patients, but it will allow us to expand in the GBA1 patient population and also eventually the sporadic PD patient population. And as I said earlier, we believe we have preclinical data that supports all of that.
And it seems like you know a lot already about the natural history of these GDPD patients. Have there been a lot of good historical natural history studies that help you understand like what happens here in the case where they're on the ERT but they're not really getting anything else, or is that an evidence gap as well that you hope to fill?
It's a great question, Patrick. It is not an evidence gap. These are well characterized, well studied patients. There are a couple of centers in the United States that have really focused on these patients. One's in California, one's here in New England. These patients travel to the NIH to get evaluated on a regular basis. And so we think that we could capture these patients because they do travel, and as I said, they are excluded from other Parkinson's studies. And so they're also a very motivated patient population group. And so the natural history is very clear with these patients.
That's really good. I mean, one of the things that just adds a challenge to many rare and ultra rare diseases is when that isn't in place. And you see the opposite when the NIH or other groups or patient advocacy groups sometimes have been investing in this sometimes for decades. It can make your job that much easier, right? Because you can really understand the endpoints, good statistical design, because you know exactly what you see in the untreated population, right?
Absolutely. And that's why this is another reason this makes the GDPD patients a great first study. I believe this is going to be a great product for these patients, but also the gateway to other Parkinsonian disease.
Yeah, fascinating. You have, I think you've presented at the past at some recent autophagy keystone conferences. I think you're presenting at an upcoming one that you're working on other targeted ligands around the autophagy pathways. Maybe you can talk a little bit about that as well, because TRPML1 is the lead program, but you've got others, it sounds like, coming behind it.
Oh, thanks for asking that question, Patrick. We've invested a lot in understanding this pathway. TRPML1 is a lysosomal target. And so that's really at the sort of end of the pathway, if you will. It's at the level of the lysosome, which is sort of the very end of the autophagy pathway, as we discussed earlier. But one of the things that we attempted to do here at Casma was identify the key autophagy genes to help initiate autophagy. So you can imagine can be really important. If you have a dysfunctional mitochondria or a dysfunctional lipid, and you want to augment autophagy in these patients specific to those targets, it would be great if you could bring the autophagy machinery to that target.
Right.
To do that, you have to understand what the key autophagy initiation genes are. So we've done that here at Casma, and we've created these heterobifunctional molecules. One side hits these key autophagy initiation genes. The other side latches onto the target. So this would be the autophagy equivalent, if you will, to the discovery of the E3 ligases cereblon or VHL in the PROTAC world, or the E3 ligase proteasome degradation world. And we have not only identified those genes, therefore, but we've discovered the ligands for these things. So this is pretty important for the autophagy initiation field. But I'll add one other part of this, Patrick, and that is, as an offshoot of that, as an offshoot of having discovered these key autophagy initiation genes and being able to drug them, we've been able to create a series of molecules that upregulates autophagy globally in a cell, but it does this without touching the mTOR pathway. So this is pretty important. People have been looking for this for at least 2 decades. Everybody knows that everybody in the field knows that you can turn on autophagy if you inhibit mTOR. Okay, but you don't really want to do that on a regular basis to a patient because mTOR is involved in so many other processes there. And so people have said, is there a way to turn on autophagy without touching mTOR? We think we have that way. We've done a number of preclinical experiments there, and we're moving into animal models here, trying to turn on autophagy, and degrade things like tau aggregates, as an example.
I don't know much about the field. What happens if you do inhibit mTOR and do it the the hammer and a nail way? I suspect lots of bad things, but what does happen?
So mTOR is involved in a whole bunch of different processes. It's involved in inflammatory processes, other immunological disorders. People, if you take rapamycin, for example, you'll inhibit mTOR. But if you take too much rapamycin, you'll have all sorts of cardiac and metabolic issues associated with that. So you can't really do that clinically. But if you can turn on autophagy, but what is known is that if you turn on autophagy, without touching mTOR, and people have done this genetically, in genetic animal models, you have things like extended lifespan. If you cross autophagy upregulated animals with Alzheimer's models, those animals won't develop Alzheimer's. So it's a pretty important area. We're not working on that so much right now because we're focused on our TRPM1 program. That program is a little bit earlier, but I'm glad you asked about it because I did want to mention it.
And in your TRPML1 and these other programs, this is all small molecules, right? So you have,
Right.
You're not playing on quite the hard mode that gene therapy and other next generation therapies are playing on. Maybe you can talk a little bit about the pros and cons of taking that approach.
So, obviously TRPM1's a small molecule. We have molecules that easily get into the brain, which is critically important here, of course. But even our heterobifunctional selective autophagy molecules are relatively small. In fact, they're smaller than the current sort of E3 ligase proteasome heterobifunctional molecules that have already been shown by other companies to get into the brain. So we're pretty, and our autophagy enhancer molecule is also a small molecule. For us, it just eliminates the hurdle of having to bring a large molecule like an antibody or a gene therapy program into the brain.
Yeah, that makes sense. You, as I mentioned at the top of the show, prior to joining Casma as COO and now CEO, you spent a long time on the investment side of the house. I'd love to hear first about your approach to risk. When you're an investor, you have a portfolio and you can have some very big swings and you can have some science that just doesn't work out. You can have that in a large pharma as well to some extent. You don't have as many degrees of freedom in a biotech though, and it's you and your team's life, right? You're spending 5-10 years on this, so you have probably a different approach to risk. I'd love to just learn a little bit about how you think about that and how you approach it, having spent time in each of these different regimes.
So that's an excellent question, and it's very true. Investors take a portfolio approach to risk, right? They're not afraid of risk. It's a matter of understanding the level of risk that you're taking and managing that. I will say though that, and that TRPML1 in many ways is a portfolio of programs, right? Because it's not just GD, it's not just Gaucher's disease, it's not just GBA, it's not just sporadic. It could be beneficial in programs outside the CNS. And so that being said, how best to manage risk? I think twofold. One, always maintain a high level of science in the company. We have put a lot of effort into understanding this entire pathway, but specifically understanding how TRPML1 works. And as I said earlier, we published on that in Science Advances a few years back. So really maintaining always a high rigor of science there. The second is the other thing I touched upon earlier, and that is, especially true in Parkinson's, you can't just have one model that works. You have to have a number of models that work because no one model really really captures the disease. And so we feel like having 4 in vivo models that have all shown efficacy and truly understanding the biology of TRPM1, those are the 2 ways that we've managed risk in the TRPM1 program.
Yeah, that makes sense. And what about your views on the market today more broadly? There's been biotech booms and busts over the last few decades, and we're in a challenging time right now, but companies like yours and others are marching forward. I'd love to just hear your perspectives on that.
So, yeah, there's no question that it's a difficult time for biotech, especially small private biotechs like Casma. The best way to, I think, manage that process is to have extremely supportive investors. And we've been lucky at Casma is that we've had very supportive marquee name investors. The company was incubated by Third Rock Ventures. The Column Group led our Series B round. So we've had very supportive investors. The way you keep supportive investors and keep them being supportive of what you're doing is the answer to your previous questions: a high level of science, a high level of rigor, which we've had here at Casma, and really sort of looking at the entire landscape of animal models, especially in a disease like Parkinson's. And so I think we've been lucky in that regard, in that we have supportive investors, but we've kept them supportive by the approach that we've taken.
Are investors as interested, more or less these days, in rare diseases? I think GDPD is a rare disease, although you made the point that you've got a platform that can go lots of directions. But right now, probably the first thing they're underwriting is the opportunity to bring transformative medicine to market in a relatively small population with unmet need. I'm curious to get your perspective on that.
So I think investors are still care about rare diseases, and certainly we're getting a lot of good investor input on that. They definitely care about rare diseases, especially if you're going to make a significant difference in the lives of those patients. They also obviously care about, diseases like GBA and sporadic Parkinson's. I'll add that GBA is 20% approximately of all Parkinson's patients. And so it's not really a tiny indication. In the United States, that's 200,000 patients. But I think the best way to talk about this is if you have a plan that says you're going to de-risk something like GBA or sporadic Parkinson's with this rare genetic disease, GDPD, I think that is really getting traction among investors, because they'll say to themselves, wow, that's an approach to de-risk, a clinical program in sporadic PD, which is very expensive. But this de-risks the program. So I think they love that. But they also say if it just works in GDPD, that's also a winner, because that's such an unmet medical need, and there are a lot of those patients. There are a fair number of those patients, even though it's a rare disease.
Yeah, that's interesting. Are there some analogs in the ones that are coming to mind for me are, for example, you've got forms of Alzheimer's or dementia and Down syndrome, for example, and then you've got the larger population. Maybe there are rare and ultra rare liver diseases that could ladder up to something like MASH that you mentioned before. How do you think about some of these other areas where you can run that same approach?
So I think that there are the biotech world has already been doing that. They've been doing that in MASH, right? I mean, there are companies that have gone after sort of subsets of those MASH patients before they've gone into, the broader MASH population, as an example. So I think that that's sort of a tried and true approach there. And certainly it's been the approach a little bit in oncology, right? To go after subsets of patients before you go into sort of these broader, indications. And I think that that's just, fortuitous in our industry, and certainly for us in the CNS. I think that the CNS is getting to be more and more of a precision medicine place, Patrick. People are focused now on what you said, things like Down syndrome patients who have Alzheimer's disease. I think almost every Down syndrome patient, as they approach their late 40s and 50s, will develop Alzheimer's patients. And I've heard of many folks that are sort of focused on those clinical programs there. It's actually an area that I also think TRPM1 agonism could be really important, and that would be an interesting and important patient population to go after with TRPM1.
Yeah.
It's one of the many reasons we're pretty excited about this program.
Yeah, I mean, you have the classic challenge of you have like a platform biology sort of approach, right? Where it's
Exactly.
You can take it a lot of different directions, and like you said, you have a portfolio in a way that you've got to make trade-offs in what you go after now and what waits to the future.
Mm-hmm. Exactly.
Well, Frank, this was amazing. I learned a lot. Is there any way that the listeners can help? Are you looking for collaborators? Are you hiring in any kinds of roles? And we'll make sure we direct people to your website. But anything you'd like to call out, now is a good time.
So I would like to call out that we are talking to potential partners right now for our TRPM01 program. We certainly can take it forward in GDPD, but once you get into these larger indications, you're definitely going to need some help from partners. And so we're definitely in the market looking for partners for our TRPM01 program, right now. And so that's the thing I would call out here. The other thing I would say is, we're pretty excited because our TRPM01 program could work across a number of genetic defects in GD, GBA, sporadic PD, etc. We also think that down the road, our program could be synergistic with drugs that actually attack things like LRRK2 or ATP13A2.
Yeah, maybe you could say more of that. How does the synergy work? Because you've got one that attacks the target directly, and another that helps to smooth the path in a way.
So I think that as an example, if you have a program that's sort of specific to LRRK2, so patients that have that LRRK2 mutation or TMEM175, they're going to work on that mutation, but those patients still have dysfunctional lysosomes.
Yeah, interesting.
Right? And so being able to sort of help those patients at the lysosomal level, in addition to the specific genetic defect like a LRRK2 or TMEM175, I think is going to be extremely important.
Yeah. Your comment that you made earlier about doubling the lifespan of mice, I know that's obviously not an analog to humans, but you also spoke about rapamycin and mTOR inhibition, which I know the longevity people love, but it had me thinking about whether is there something interesting to explore here around more general biomarkers of longevity. This is such a key pathway that it just has me thinking about that.
Yeah, it comes up all the time, Patrick. We're not really focused on longevity. We're focused on neurodegeneration, and we're focused on clearing dysfunctional organelles like mitochondria or tau aggregates or lipid. So we're focused on really sort of more diseases of aging as opposed to aging itself. But I think what's clear is that the autophagy pathway plays an important role in cellular homeostasis, right? And it has a relationship to things like senescence, which people are looking at on the longevity side. So we do get sort of inbound interest, if you will, from people who are interested in longevity, because if you augment autophagy, it leads to increased lifespan, and not just increased lifespan, but increased healthy lifespan. We're not focused there. We're more focused on those diseases of aging. But I suspect that this is going to be an important pathway, as people get more and more interested in longevity and, wellness and these kinds of things.
Yeah, amazing. Well, Frank, thank you so much for sharing all this with us today, and I'll be rooting for you on all the progress in your clinical development.
Thank you, Patrick. I really appreciate you taking the time, and I'm really excited to be here at Casma and help develop this program.
Amazing. And thanks everybody, as always, for listening, and we'll see you next time.
Take care.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.